Hello and welcome back to Microbiology. Chapters 14 and 15 are really going to go together. So we're going to do 14 and then we'll have a separate narrated PowerPoint for 15. But there's going to be a lot of overlap. So let's go ahead and get started with Chapter 14, which will be host defenses. Whenever we talk about host defenses, we're really going to break it up into three different lines of defense. The first line of defense is going to be basically barriers. They're non-specific. They're really there to prevent foreign things from getting in. The second line, though, is also non-specific, but is going to have cells that are targeting and destroying pathogens as they get in past the barriers. There are also going to be very large system-wide events like inflammation, fever, even things like complement that we'll discuss as part of the second line. But again, both the first and second line are non-specific, and really the topic for this chapter. The third line, on the other hand, is what we would call specific or adaptive immunity. And whenever I say specific, it's illustrating that we're going to be targeting a very specific pathogen. So we're not trying to kill everything, we're just trying to kill one specific thing, like maybe E. coli, or staph, or strep. The illustration here is, again, these cells in the third line target one specific thing for removal. The other point of this, though, is since it's adaptive, the levels of protection change over time and will provide long-term immunity. This is a beautiful thing that really helps protect us from infection over multiple years. So here's a visual representation of exactly what we just talked about. The non-specific or innate immunity is really split into the first and second lines. Again, these are non-specific features. Again, that first line is going to be your physical barriers or your chemical barriers. But again, both of these things are barriers trying to prevent things from getting in. The second line comes in whenever things do get in. But again, since this is non-specific, this is normal, non-specific response to remove the threats, including phagocytosis, inflammation, fever, and then antimicrobial proteins like complement. The third line is going to be acquired or specific immunity. And the idea is these are, again, specific white blood cells that are B and T cells. Their job is to target very specific pathogens and remove them. Whenever we talk about this chapter, it's important to understand that we're just going to really focus on these first two lines, the non-specific immunity. We will briefly discuss specific, but again, chapter 15 will be focused on specific immunity. So let's start going through that first line of defense, which is the physical and chemical barriers. There are lots of these things. And this image is illustrating several of the techniques that you may have never thought about as being an actual physical or chemical barrier or defense. Things like even the act of urination allows for the removal of pathogens. We can flush out the urethra whenever we urinate. That is a barrier. We're creating a barrier to prevent entry. Again, the skin is a wonderful example of a barrier. The idea that we have intact skin that is routinely shed allows for a physical barrier to the outside world. Examples of chemical barriers include things like low pH of the skin, or even the sweat that can wash away microbes, or even then deposit things like salts on the surface of the skin. The idea here is you have lots of physical and chemical barriers in place, and they work all the time. They're not just working on specific pathogens, but all pathogens. The reality is with physical and chemical defenses, we're not specific, we're just a general purpose defense barrier. So let's talk more about the skin, because the skin is probably one of the most important organs that we have in terms of protecting us from infection. The skin structure in itself is important because since it's epithelial cells that are compacted in multiple layers and basically put together with keratin holding everything together, it makes a pretty impervious barrier that is also waterproof. And as a result, very few bacteria can actually get through skin. Other barriers could include things like hair follicles and sweat glands, but each of these do offer some protection. Hair follicles are a common source of things like oils, and as a result, again, lower pH in these environments. Sweat glands help produce sweat, but even then help wash away or push away microbes and also deposit things like salt. For everything that basically is a hole that opens up to the outside world or is connected to a hole that opens up to the outside world, we will see mucous membranes. So mucous membranes line things like the digestive tract, the urinary tract, the respiratory tract, the eyes, basically every hole you have. The idea is mucus is how we try to protect these pathways. That mucus is going to create basically a barrier. And the idea is that that mucus can also capture up and hold the microbes. But I should really point something out to you, that mucus is great at grabbing the microbes, but also provides potentially a source of energy for the microbes. And as a result, if we don't move the mucus out, we could have basically infections start to develop. The respiratory pathway, again, has mechanisms to help try to push the mucus out. So there are events, there are structures like the ciliated cells and microvilli that help us move mucus. Events like coughing also try to push mucus out. And so the idea here is we have all these mechanisms to really push mucus. And as long as mucus gets pushed out, we are protected. But if mucus gets trapped, now we have a risk of infection. Genitourinary tract is another pathway or portal into the body. And as a result, we need to protect it against pathogens. One of the best protective methods we have is basically urination. By emptying the bladder, we can flush out the urethra and any pathogens that are trying to make their way up it. And this is very important to do. So it's very necessary that you take time to urinate if you need to urinate. And that, again, after things like intercourse, urination becomes very, very necessary, as things that would usually not be associated with the genitals may be introduced. So urination is a good protective strategy that our body uses against UTIs. Another important thing to note in terms of protection against pathogens is the presence of other bacteria that are not there to hurt you. Again, these microbial antagonists will prevent typically pathogens from living in the space because there's not enough resources, and typically it may not be a favorable environment for the pathogen. So again, having good bacteria or other microorganisms in and on you help protect you. We have non-specific chemical defenses in places like the skin and mucous membranes that help protect us against pathogens. The lysozyme is one of the better examples that we have. A lysozyme is an enzyme found in tears and saliva that actually break down peptidoglycan by hydrolyzing it. The idea is it's non-specific because it attacks any bacteria that uses peptidoglycan, not just one type. Things like lactic acid and electrolyte concentration in sweat also affect lots of different microbes. There are other non-specific chemical defenses, including things like the stomach. The stomach is really just a bunch of acid, and as a result, again, we can break down pathogens as they come in. In the intestinal tract, we see things like the digestive juices and even bile. And believe it or not, there's actually lots of antimicrobial components as part of semen, and it's because one of the parts of the ejaculatory event is we bring down the pH of the vagina, making the vagina at risk for infection. So semen also needs antimicrobial compounds to help protect the vagina long enough so that there isn't an infection that occurs. And if the body has to choose between a newly fertilized egg and an infection, it'll always ditch the egg to fight the infection to keep the host alive. The idea here again is if we're going to lower pH in the vagina, we also have to protect the female anatomy long enough for, again, pH to return to normal. Immunology is going to be the study of all the features that are the second and third lines of defense. Really what this is going to be is the study of a lot of white blood cells. So what we're going to do whenever we talk about a lot of immunology is talk a lot about white blood cells. And the focus of the immune system here is really to survey the body, find foreign things, and destroy them. In this image, notice how we have what should happen whenever our white blood cells encounter our own cells. Notice there should be no reaction. But whenever a white blood cell runs into a foreign entity, there should be a reaction. And in this case, this is a phagocyte that will consume the pathogen and destroy it. So how do we recognize self and non-self? Well, really we do this by using markers. Markers are molecules on the surface of a cell. They're going to be composed typically of proteins and sugars, and really are our targets to be evaluated by the immune system. So one of the things that we should talk about is the mononuclear phagocyte system, or MPS. the MPS is going to be very important in helping to control the risk of infection. This is also going to be a primary concern in the extracellular space or extracellular fluid around the cells, as this is a place that routinely pathogens would love to grow in. The bloodstream is another place of great concern, and as such, we often keep a lot of white blood cells there, as well as the lymphatic system, which is our return system from the extracellular space back to circulation. So you can see exactly what we're talking about here. Notice we have the cells of our body, and then we have circulation for our body. But notice we have this space in between, and in this space we dump resources to help feed and keep the cells alive. But since we have all these resources, it is also a wonderful place for pathogens to grow. So we like to keep a lot of white blood cells, like dendritic cells, hanging out in this space to eliminate those threats. Resources can and will be picked up from this space and carried back to circulation through the lymphatic system. So we'll often see these lymphatic capillary beds that are going to help pick up the lymphatics, basically the fluids and whatever's in it, and carry it back towards circulation. So let's talk about the common flow of events that happens whenever a pathogen comes in through the skin. Whenever we see it, they typically end up in the extracellular space or reticular tissues. That then can lead to being picked up by the lymphatic circulation, which ultimately carries the resources back towards the blood at the heart. So notice we're showing you kind of where we see the hot spots for the mononuclear phagocyte system, or MPS. There's some common features that you should note. If you look, where the highest concentrations, or where the purplest regions are all those pathways into the body. And so the idea is we know that lots of pathogens will come in through the respiratory pathway, come in through ingestion and digestive processes, or even through the genitals. The idea here is we're going to, with the MPS, keep white blood cells, phagocytes, in close proximity where pathogens will come in. We have associated lymphoid tissues that will be kind of areas where we have large amounts of MPS. The idea here is we'll have these things in places where we see the greatest risk of pathogens being present. Really, the major function of the lymphatic system is to provide a pathway back to circulation. And so this is basically an opportunity to have a drain-off system for inflammation. So whenever all the fluids again rush in during inflammation, we have pathways to take that fluid back around into circulation. And in doing so, we also move potentially pathogens. But another important thing to note is we're also going to move things like white blood cells and even antibodies, allowing us then to protect lots of areas. So lymphatic fluid is going to be made up of primarily lymph, which is kind of like plasma. Plasma, again, being primarily water-based, but it'll also have dissolved resources in it. The lymphatic vessels are actually going to be located all throughout the body, and basically are set up in such a way that extracellular fluids can easily be picked up into that pathway. But there is some major differences between this and circulation. With the extracellular pathway, the lymphatic vessels, we're going to see the resources are going to be pushed not by an organ like the heart, but rather by the movement of the individual. So skeletal muscle contraction is actually what pushes lymph. The idea, though, is that lymph, if we weren't moving, would settle back out. So another important fact about the lymphatic vessels is they have basically one-way valves, which allows for resources to pass through, but then they can't typically leak back down. So you can see here, this is the comparison between the circulatory system and the lymphatic system. Notice again we have ample amounts of lymphatic vessels, and notice that they all start towards the extremities and push their way back towards the heart. And the heart is again where we would see this reintroduction back into circulation. So we move back towards the heart where we can have that reuptake into circulation. I want to point out some things that along the way we have these protective structures that we call lymph nodes, located in places like the groin, armpits, and even around the head and neck. The idea is these pathways, the lymphatic pathways, will lead through these nodes that allow for screening to occur, that allows us to introduce potential pathogens to the anti-pathogen cells, the white blood cells. And so really these are primarily going to be made of your B and T cells, your specific cells that hang out in lymph nodes, and as a result we have a chance to activate the specific immunity. So there are again several organs and tissues that will be associated with the lymphoid system, including things like the thymus. The thymus is going to be very well known for helping to mature T cells, which are a part of the specific immunity. We also see the lymph nodes, which I just described a second ago as being the basically meet and greet areas where potential pathogens or even our phagocytes can introduce our B and T cells to potential pathogens. The spleen is also going to be an important factor because the spleen helps filter out parts of blood, including any pathogens that may have made its way into blood. And then we have our associated lymphoid tissues that will be located in places that will allow us to help protect the body where we need the most protection. So here's the thymus. As you can see, it actually is a very large structure early in life. And it's because it serves its greatest purpose very early as we're trying to produce our T cells. The idea though is over time we need less and less of these T cells produced because we already have a robust set. And as a result, the thymus actually physically shrinks over time. Lymph nodes, as I've already described, are going to be small bean-shaped organs that will be located in regions like the groin, the armpits, and the neck. And ultimately, it's a way to kind of filter materials, but also expose B and T cells and specific immunity to pathogens or white blood cells that have encountered pathogens. The spleen, as I mentioned before, is very important in basically helping filter out things from the blood, like worn out red blood cells. But they even will pull out pathogens or even white blood cells. And as a result, again, we often have a lot of white blood cells hanging out to destroy the pathogens that are pulled from blood. And then we do have, again, those miscellaneous lymphoid tissues, things like the tonsils, breasts of a pregnant woman become, again, a very big focus point for white blood cells, especially during lactation, as we don't want to pass specifically bad bacteria to the offspring. We also see galt, malt, balt, and salts, again, all these associated lymphoid tissues, and even Peyer's patches that help in establishing protection in places where we might need more protection. So, whenever we talk about blood, I'm not going to really spend much time or expect much out of you whenever it talks about whole blood. You should have this information already been presented to you in places like anatomy and physiology. Here, we're not going to focus much on what is whole blood. Instead, we're going to focus more on certain parts of the blood, including features like white blood cells. So we should talk a minute about production of blood because all blood is produced the same way, including white blood cells. Early on in development, we do not have the organs necessary for making blood through, again, hematopoiesis, and so we use the yolk sac as being the primary source for blood production. But even later in the embryo's production or development, we see the production of blood transferred to the liver. But as the fetus starts to develop a hard skeleton, we see the production of the blood gets transferred to, again, the bone marrow. And from the rest of that point on through the rest of your life, again, the bone, specifically the bone marrow, tends to be the source of blood production. So let's talk about that hematopoiesis procedure because, again, it starts off with a stem cell we call the hematopoietic stem cell. This is the precursor to all blood cells and ultimately leads to, again, all these different undifferentiated cells that then become our specialized cells. So let's show you this process. So again, everything starts off with the hematopoietic stem cell found in the bone marrow. And from there we get these unspecialized cells that do become more specialized cells over time. The idea is, again, all blood is produced this way. Red blood cells, platelets, and all the white blood cells. I already told you I don't care much about the red blood cells and platelets, stuff like that. What I care about in this class will be our white blood cells. The white blood cells are what affect things like pathogens, and that's why we're concerned with them. So let's go through them now. The first thing I want to draw your attention to is the production of the lymphoid stem cell that turns into our big lymphocytes. These two lymphocytes are our specific immune cells, the B cells and the T cells. Again, B and T cells are specific. Notice there is an offshoot called the NK or natural killer cell. The NK cell is going to be like a T cell, except for it lacks a lot of the specificity. So this is not as specific as the T cell was, and as a result we consider this to be a non-specific cell. So let's then focus on the other versions of white blood cells by looking at the other pathway. So this is the stem cell for all blood cells except for lymphocytes. So we see things like the monoblast that produces the monocyte. The monocyte is going to be only found in circulation, because once it leaves circulation, it grows very, very large and turns into a macrophage. Again, an easy way to remember this is because it's macro or large, and it's a phage, which means it eats things. This guy cruises around and eats foreign entities. There are specialized versions of macrophages. Again, there are several of them, but one of the more common ones we'll talk about is the dendritic cell, which is commonly found associated with tissues close to the skin. And as a result, again, this guy will play a huge role in trying to find and eliminate any threats. Another important feature of this dendritic cell is it does present antigens quite well. Macrophages can also present antigens, which we'll talk again more about later, but the idea is these two cell types help eat things, but then also activate specific immunity like B and T cells. So let's look at the myeloblast, because the myeloblast can really help us develop all the other cell types that we're going to be concerned with. Myeloblasts turn into things like neutrophils, basophils, eosinophils, and mast cells. For our purposes, we're going to consider the basophil and the mast cell to be basically the same. Okay? We're just going to say they're part of inflammatory events. But I do want to draw your attention to these two smaller boxes, the granulocytes and the agranulocytes. Granulocytes are called granulocytes because they have granules inside. Agranulocytes are not going to have granules, as you can see here. And so really, let's talk about the three granulocytes here. Neutrophils are going to be phagocytes found in the blood that readily focus on killing bacteria. And this is important because if we see elevated levels of neutrophils, this suggests that there's a bacterial infection. Basophils usually are not super abundant because they're going to help activate the events of inflammation through things like chemicals released. Eosinophils are going to be very important for eukaryotic infections. So if we see elevated levels of eosinophils, again, that's a good indicator of a eukaryotic infection. Another name for white blood cells is, again, leukocytes, so make sure that you're aware of that whenever it comes time for testing purposes. And we're going to spend more time talking about granulocytes and agranulocytes in the upcoming slides. So let's start off with granulocytes. Granulocytes are really going to be made up of neutrophils, eosinophils, and basophils. Again, they're called granulocytes because whenever we stain them with eosin or methylene blue, we see the presence of granules. These granules are not consistent, though, across the different types of cells. Each cell will have a different use for their granules. Neutrophils are going to be a major component of the circulating white blood cells, and really typically they produce toxic chemicals and do phagocytosis. Eosinophils will be much less common, and as a result, again, won't be a prominent cell whenever you do a lot of basically stains of blood. I do want to point out that they have pretty varied targets. Whenever we talked about neutrophils, it was really just bacteria, but eosinophils can target a variety of different eukaryotic things, including very large eukaryotic pathogens like worms. As a result, they need to be rather diverse in the ways that they can attack. Basophils, again, are also very rare, so we see them being around 0.5% of circulating white blood cells. And ultimately, they're going to be very similar to mast cells and help control things like inflammation. Agranulocytes are going to be, again, without granules. So whenever we stain them, we don't see the granules. And we really turn this into two different categories. So agranulocytes are classified as either lymphocytes or monocytes. The lymphocytes were, again, that third line of defense, the specific cells. And really, they're just B and T cells. And they're going to be roughly 20 to 35% of circulating white blood cells. And whenever we see, or a cool feature that we see here with these lymphocytes, is they become more numerous over time. If you recall, the specific immunity was also called adaptive because it changes as we need. And really, what's cool about it is we develop memory. And so the more exposed we are to a pathogen, the more cells we hold around for the next future encounter. So by the time you're at your height of immunity, the lymphocytes become incredibly common. Lymphocytes, again, were B and T cells. So let's talk a little bit about B cells. B cells are going to fight through a process that produces antibodies. We call this type of fighting humoral immunity. Whenever we talk about T cells are actually going to fight by going to the site of infection and killing cells. So we call this cell-mediated immunity. B cells are going to, again, produce antibodies, but the form of B cell that produces antibodies we call a plasma cell. And it's important for you to understand that antibodies are really nothing more than proteins that will simply bind to what they recognize, which is an antigen. T cells, again, on the other hand, going to the site of infection, will destroy the pathogen that they recognize, but only that thing that displays the features of the pathogen. So monocytes and macrophages were the other half of, again, the agranulocytes. And so with monocytes, we see these things being very large white blood cells and being found in circulation. But whenever they leave circulation, they actually get even larger and become macrophages. Macrophages are long-lived and capable of multiplication. I do want to point out that these are very versatile because they're nonspecific and do a lot of surveying environments outside of circulation, so they're very necessary. Ultimately, the macrophages are going to clean things up. They're going to clean up those messes through phagocytosis, so they can clean up things like the remnants of inflammation and even infection. The idea is they'll cruise around in the environments outside of circulation and fix any issues in terms of waste products from events like infection and inflammation. A specialized version of the macrophage is the dendritic cell. We call this a dendritic cell because it has these long extensions on their surface. The idea here is these dendrites increase surface area. And the reason that's relevant here is because another important feature of macrophages is their ability to present antigens to B and T cells. The idea here is that they can help activate ultimately forms of immunity. And so we need surface area to present those antigens on. We'll talk a lot more about antigen presentation in the next chapter. Again, erythrocytes and platelets I really don't care much about for the purposes of this class. You should again have worked on this whenever you guys were going through anatomy and physiology. So phagocytes are, again, general category of cells that consume other cells. The idea here is that they're going to survey the tissues, find the threat, and then dissolve or break down the threat after it's been ingested. So naturally, we need to be able to recognize what are threats. So they recognize features called antigens and foreign matter. So for now on, we're going to refer to, again, foreign features as being antigenic features. We should talk a little more about neutrophils and eosinophils. A pretty important thing to understand about neutrophils is they're going to play a very big role in early inflammation response. The idea is whenever we have the potential of infection coming in, one of the most abundant things that we have to fight the infection is neutrophils, and as a result they tend to be the first ones on the scene. Eosinophils, though, are going to be very attracted to things like eukaryotic infections, and as a result their primary association is to parasitic infections. And so they come in and help try to remove the parasites. But since many of the parasites are eukaryotic and as big as the cell is itself, we often don't do a lot of phagocytosis, but instead bombard the other cell with chemicals. So here's that illustration of the monocytes and macrophages. Again, in circulation we refer to the cells as monocytes. But once we see it move from blood into out of circulation into the tissues, we call it a macrophage. So here we can do some comparison of macrophages. So this is an alveolar macrophage associated with the alveoli in the respiratory pathway, and notice how it is consuming bacteria. It is moving itself up and around the bacteria. They're taking it in to ultimately break it down. And the fancy word that we have for the unique macrophages is histiocytes. A histiocyte is again a specialized macrophage that goes to a very specific tissue. Alveolar macrophages, as I just talked about, were found in the alveoli, associated with the respiratory pathway. Kupffer cells are routinely found in the liver. Dendritic cells are associated with the skin. And we also see lots of macrophages occupying things like the spleen, lymph nodes, bone marrow, kidneys, bone, and even brain. So let's talk more about phagocytosis. We haven't really done much with it yet, so let's go through it. Because in order for phagocytosis to occur, we need to have receptors that allow us to recognize foreign things, which we're again calling antigens now. The idea is there's some antigens that are clearly belonging to something foreign. We refer to a lot of these very clear antigens that do not belong as PAMPs, or pathogen-associated molecular patterns. These are features we recognize as being part of a foreign entity like bacteria. A good example of a PAMP would include something like peptidoglycan. We never produce peptidoglycan, so whenever we see it, we know that belongs to bacteria. And so again, we will have receptors that will look for things like that. That allows for the binding to and attachment of the white blood cell, the macrophage here, to the pathogen. And as a result, we gather them up and we pull them inside in a vesicle we call the phagosome. But in the phagosome, the bacteria don't die. The only time they will die is if we bring components known as the lysosome in and fuse it with the phagosome, forming the phagolysosome. Once the phagolysosome is developed, enzymes and reactive oxygen products will be introduced to the pathogens, killing them and destroying them. And then we'll dump the waste out to typically be recycled. So let's talk about chemotaxis and ingestion. Again, the movement is into response to chemicals. And again, I told you that there are certain patterns that we recognize as being pathogens. We call them pathogen-associated molecular patterns, or PAMPs. Again, we recognize this as being not part of us, and as a result, we will attack it typically on site. So things like peptidoglycan, LPS or lipopolysaccharides, gram-negatives, even the presence of double-stranded RNA, because again, we don't produce double-stranded RNA. But I told you we just simply had receptors to find and bind to those PAMPs, but they have special names. They're PRRs, or pattern recognition receptors. PRRs, or pattern recognition receptors, recognize and bind to PAMPs, which were pathogen-associated molecular patterns. Ultimately, PAMPs tie to PRRs and allow for that integration that allows for then ingestion through phagocytosis. But let's talk about another unique receptor called a toll-like receptor, or TLR. This is a type of PRR, or pattern recognition receptor that recognizes PAMPs. But this does not typically then want to allow for the ingestion of the microbe, but instead releases basically a chemical cascade that sends information to lots of cells at the same time. So notice here, whenever the toll-like receptor is not activated, they're apart. But whenever the foreign entity comes in, the two parts come together to send a message into the cell that allows for replication of basically important chemical information to be sent out of the cell to other cells. This is a warning system. So we're taking a message and we're amplifying it and sending it where it needs to go. The phagolysosome was important for us to discuss because, again, if the lysosome never fuses with the phagosome, we will never kill the bacteria. It's only whenever the lysosome and phagosome come together do we form that kind of caustic pocket that destroys and breaks down the microorganisms, and it typically happens quite quickly, roughly around 30 minutes. One of the ways we destroy or break down things is, again, using dangerous forms of oxygen. So the idea that we can use basically things like singlet oxygen or hydroxyl free radicals to break things down would be very necessary. But if you're going to produce these bad components, you yourself should have mechanisms to help break that down so that we don't hurt our own cells. So let's go through and talk about the 4 basic or classic signs and symptoms of inflammation, including redness, warmth, swelling, and pain. There is going to be a 5th sign, and that's typically the loss of function. Ultimately, all these signs of inflammation serve as ultimately a warning that injury has taken place or potentially infection. So really, the goal of inflammation is to ultimately mobilize and bring immune cells to help try to fix the current problem. This could even again set in motion mechanisms to repair damaged tissues and remove foreign entities that have invaded. So let's go through the steps. As you can see here, nail has penetrated our skin and our defenses. As a result, on that nail, pathogens have now been introduced into the body in a place where we don't have as many defenses. So naturally, inflammation should result. One of the first things that should happen whenever we pull this nail out of the wound is blood should flow out, which helps flush the wound. But at the same time, we also see an increase in blood flow, bringing in resources to allow for the clotting, to stop the bleeding. But now we have a situation where we've walled off and stopped potentially future pathogens from getting in, but we still have a bunch inside. So we have to start attacking this by attracting things like neutrophils to the site. Neutrophils are then going to help start fighting and destroying the microbes. But in doing so, there will be casualties on both sides, and as a result we see pus forming. That pus then has to be broken down and cleared up, and as a result we often bring in things like macrophages to clean that mess up. There are going to be lots of messages that are going to be sent around, including things like vasoactive, which affects endothelial cells and smooth muscle cells of blood vessels. We also have chemotactic factors, also known as chemokines. Again, I'll use the word typically chemokines to attract white blood cells. And then we also have inflammatory mediators that can cause things like fever, the stimulation of the white blood cells like lymphocytes, and even prevent the spread of things like viruses. So here's a big list of inflammatory mediators and other cytokines. I don't really need you guys to know these. I do want to draw your attention to one in particular, and that is the interferons. Interferons are actually going to be an important message that helps inhibit viral replication. And so the idea here is that whenever we're being attacked by viruses, our cells can release interferons that help promote other cells to protect themselves, decreasing the risk of infection. Why this is so relevant here is this idea that interferons are non-specific. They're not released just for one virus type, but for any virus. So if we start actively putting interferons into you, your body would think it's under attack by viruses and try to fight back. So this could be a technique to fight viruses in the future. That swelling or edema is really caused by the leakage of resources into the tissue. It's important to understand that swelling is that overaccumulation of those fluids. And those fluids bring with it heat and ultimately swelling. That pressure again can create pain, which we again associate as being parts of inflammation. One of the important things that must happen to allow all this migration of resources is basically the separation between the cells associated with the circulatory pathway, so the endothelial cells. Ultimately, what we see is a movement of white blood cells from circulation to the tissue. There's a special name for this, and that's diapetesis. Diapetesis is another form of chemotaxis. There's chemical factors encouraging these white blood cells to show up here. Ultimately, we point out that edema allows for resources to flood in that can even dilute things like toxic substances. It also helps form things like clots and brings the white blood cells to help destroy the foreign entities or dead tissues. Late in inflammation, though, we do see the attraction of things like your lymphocytes and macrophages. The idea is that macrophages should come in and clean up any messes in terms of debris that's left behind, and then the lymphocytes will make sure and destroy anything that is still remaining in terms of the foreign entity. So T cells would come in and kill the intruders directly, while B cells, we would see the flooding in of antibodies to do the attacks. The next major event that we should talk about is fever. Fever is an increase in body temperature that is going to be typically a pretty good indication of infection. There are really three levels of fever: low grade, moderate, and high. Low grade tends to run up to around 101 degrees Fahrenheit. Moderate falls between 102 and 103, and then high would be 104 or higher. The idea is fever is being done to help fight infection, and as a general understanding, fever tends to be helpful, as long as it tends to be on the lower end of the spectrum. High fever should always be treated and addressed, because high fever is now potentially dangerous to the person involved. So what can help control things like fever? Well, pyrogens. Pyrogens reset the hypothalamic thermostat to a higher setting, again raising the body temperature. So what can cause these things to happen? Well, they can be exogenous or endogenous. Exogenous examples include things like the presence of toxins, or even things like the introduction of vaccines. Endogenous include, again, chemical messages, things like interleukin and tumor necrosis factor, as well as, again, chemicals being released by monocytes, neutrophils, and macrophages. As I said before, fever is going to be very necessary in preventing viral replication and infection. One of the things that I want to point out is, yes, it will kill some of the more temperature-sensitive microorganisms, but another big part of this is stopping the availability of iron. Without the iron, many microbes can't do a lot of metabolism. And we basically take all the iron and allows for an increase of our own metabolism that allows for more immune response. So again, treatment for fever is something that is highly argued. Overall, fever should be helpful as long as it's not high or prolonged. Okay, so the idea here is higher prolonged fevers are dangerous and should be treated. Antimicrobial proteins do also exist. I already pointed out to you again interferons. Interferons are going to be necessary in helping protect against viral infection and even cancers. So this is again a defensive mechanism that we can use to help fight. So you can see here the interferon is again not specific. So as a virus enters a cell, one of the last things that the host is going to do is to produce interferon information to release a chemical, the interferon, to inform the neighboring cells that there is a virus nearby. So giving it time to produce antiviral components that basically stops the virus from causing problems. Another important antimicrobial protein is complement. Complement is a very big and complicated system, so we're going to keep it simple. Complement is going to be a series of proteins that are always found in the blood, but they're usually not working together. The idea is they must be activated through a cascade that allows them to assemble and start attacking cells. There are multiple pathways that allow for this process to happen, including the classical pathway, the lectin pathway, and the alternative pathway. The classical pathway is named as such because it was one of the first discovered, where an antibody binds to a microbial cell and promotes then the activation of complement. The lectin pathway can be activated by mannins, and the alternative pathway is activated by bacterial or fungal cell walls, viruses, or parasite surfaces. The idea is the microbe itself activates it. So if you notice here though, all these should end in the same way, which is the assembly of a bunch of different proteins to produce a structure that penetrates the cell involved. And so the idea is these proteins assemble to make what we call a membrane attack complex. So you again can see all three pathways end in the same basic response, which is the formation of those proteins that make the membrane attack complex. The antimicrobial proteins also exist out there. These are typically going to be very small peptide chains and will interact typically with the bacterial membranes. Causing disruption in the membrane can result in death of the cell.